everyone, and welcome back as we cover Chapter 6, Cellular Respiration, Obtaining Energy from Food. This is one of the more complex topics that we will go over this semester, so make sure you read the book chapter a couple of times, especially if this is the first time that you're learning this material, and review the PowerPoint several times as well. If you don't understand it the first go around, that is normal. That's very common because, again, this is a difficult concept to grasp. So repetition is key here for any difficult material, but especially for this chapter and also the photosynthesis chapter. That is what I recommend in order for you to really learn the material. That's what I have found helpful for students in the past. By the end of this chapter, we will be able to answer the question, why do we require oxygen to live? So we all know that we need oxygen in order to survive, but have you ever thought about why? And usually when I ask this question, I will get responses like, well, we need it for our cells. And yes, that is true, but why do we need it for our cells? So we're going to really dive into the intricacies of human physiology, and we'll be able to answer in full this fundamental question, why do humans and many, many other organisms require oxygen to live? The next couple of chapters, we will be covering energy transformations. So overall, the processes of photosynthesis and cellular respiration are complementary. We have an initial input of solar energy coming into the Earth system from our sun, of course, and plants and other autotrophs, meaning self-feeders or organisms that can make their own food, are able to harness solar energy and transform it into a chemical energy, into the energy of glucose, which is a sugar, a monosaccharide that we learned about in Chapter 3, if you recall that. And a byproduct of that process is oxygen, molecular oxygen, which will go into feeding the process we're going to be learning about this week, cellular respiration. So the products of photosynthesis are the inputs to cellular respiration, and the products of cellular respiration, conveniently, are the inputs to photosynthesis, so that we can see this is literally a cyclical event. However, it's important to know that some energy is always going to be lost to the system as heat, and that is not harnessable. It's just a byproduct of inefficiency. So these are not 100% efficient systems, but they work for our needs, because what is really produced at the end of the day is this molecule that the cell can then use for energy called ATP. So basically, we are going to be looking at the bottom half of this circle diagram here. We're going to be looking at the inputs to cellular respiration and then the outputs of cellular respiration. And the main goal is going to be to make this energy molecule ATP. So while photosynthesis is a energy transformation going from solar to the chemical glucose, cellular respiration is also an energy transformation where we transform the chemical glucose into the chemical ATP. And then, of course, if an organism cannot make its own food, like us and other animals, they are deemed heterotroph or other feeders. Keep in mind, however, that all autotrophs are able to perform both photosynthesis and cellular respiration. So they make their own food and then break it back down for energy. And heterotrophs only do one of these processes. We can only undergo cellular respiration. So we have to eat the plants or other animals that ate plants and then break that energy down in our mitochondria. So again, the process of photosynthesis essentially will harness sunlight energy and lock it into a chemical called glucose, or this simple sugar here. So this 6-ringed carbon should be somewhat familiar to us by now because we went over it in chapter 3. And if you recall from chapter 3, glucose is the primary energy source of all cells. Now, cells can use other types of monosaccharides like fructose. Cells can also use lipids as energy sources, but the preferred and the favorite method is this glucose molecule. So for our purposes, think of glucose as the primary energy input for all cells. Cellular respiration, or the process we're going to go over today, is this gray arrow here. So how do we convert the chemical energy in glucose into another chemical called ATP? And this is the structure of ATP down here at the bottom. ATP stands for adenosine triphosphate. And of course that tri in ATP stands for 3. So we can see this very large complex molecule here. We have a nitrogenous base group. We have a sugar. And then we have 1, 2, 3 phosphate groups attached. That's where the triphosphate component of the molecule comes from. And then, of course, the organelle we are going to be concerned with for this process is our mitochondria. So when you were in middle school, your science teacher most likely honed into your brain that the mitochondria is the powerhouse of the cell, right? And that's true. But breaking that down further, that is what this means. The mitochondria is able to produce a usable form of energy that the cell needs. Cells cannot directly utilize the energy stored in glucose. It has to be modified and converted into ATP first. A really good analogy to this is crude oil versus gasoline. So to run your vehicle, you need to fuel it with gasoline. But originally, that gasoline was drilled up from the earth in the form of crude oil. Your car cannot run on crude oil. That oil needs to be refined first into a usable product. So that's basically what our cells do. What's what the mitochondria do? They take the raw form of the energy, glucose, and refine it into a usable form we call ATP. In terms of the word respiration, you may have heard that, and it's another word to breathe, because if you think about what's happening when we breathe, it's nothing more than simple gas exchange. So we breathe in molecular oxygen from the air. That molecular oxygen is carried to all of our cells via our bloodstream, our red blood cells, as their primary function. And then in the cells, okay, that is where this process occurs, in the mitochondria. So this is why specifically we need oxygen for cellular respiration to occur. And we'll look at where in this process this happens a little bit later. And then after cellular respiration has occurred, after that glucose is converted into ATP, we will then breathe out carbon dioxide as a waste product, and a little bit of water is released as a waste product as well. So the term respiration means to breathe. It's a gas exchange at the whole organism level. Cellular respiration is essentially the same thing, but on the cellular level. So just a little bit of a look at the nuances between those definitions. Here's the overall equation for cellular respiration. Make sure you know it, or make sure you are able to identify it if I were to show you this image on a quiz or exam. So again, we have two basic inputs, always on the left hand of any sort of chemical reaction of the arrow. Glucose, which is a simple sugar made by plants via photosynthesis, plus some oxygen, which we breathe in from the air, which is also a product of photosynthesis. 3 stages here of cellular respiration that we'll go over. And through those processes, we will release some carbon dioxide as waste, some water as waste, and then we will make that usable form of energy called ATP. And then thinking back to that circle diagram that we looked at before, we always will lose some heat because this process is not 100% efficient. Same thing with your automobile. So once that crude oil has been refined to gasoline, a usable form, and it fuels your car to work and run, 100% of that gasoline energy cannot be harnessed by the car. A lot of that fuel inside the bonds of gasoline is lost to the system as heat as well, just like our own bodies. So, of course, if you run your vehicle for any amount of time and you were to feel the hood, it would be warm, and all of that is the heat that is lost from the inefficient use of those gasoline molecules. In our 3 stages that we will look at individually coming up, 1 is called glycolysis. 2 is called the citric acid cycle. It's also known as the Krebs cycle. And then the 3rd and final stage where the magic happens is called oxidative phosphorylation. All right, well, let's break down this process here. Stage 1, glycolysis. Again, it's helpful if you're able to break words down, and that can help you define them here in biology. So glycolysis or glycolysis literally means to break apart sugar. Glucose, that is essentially the first thing that happens outside of the mitochondria. So organisms that do not have mitochondria, like bacteria, can also do this. Okay, so you do not have to be a eukaryote in order to perform glycolysis. So just keep that in mind as well. So we're not in the mitochondria yet. We're in the cytoplasm of the cell. And there are enzymes that are just floating around in the cytoplasm that will take a molecule of glucose. So here we have our 6-carbon glucose. And it will essentially break it right down the middle. Okay, so we can see that when that 6-carbon glucose is broken into 2, some stuff happens. The first thing that happens is some of those electrons that are stripped from breaking that molecule are picked up on an electron carrier, and that's what this NADH vehicle type structure is. It's just a big molecule that will pick up those stripped electrons. Also in the process, we have some protons that are generated. So essentially the hydrogen atoms in the glucose, so C6H12O6. The H in glucose is basically what we need. We need the hydrogen atom. And if we think back to chapter 2, an atom is made of protons, neutrons, and electrons, but hydrogen is one of the smallest, or the smallest atom, with 1 proton and 1 electron. So that's all we've got here. So when we break this apart, we're essentially breaking apart some hydrogen atoms and rendering them into their electrons and protons, So that's what's happening on the right. All right, we're stripping glucose of its hydrogen atoms, and then we're breaking the hydrogen atoms into the electrons and the protons, which we see here at the bottom. The electrons are picked up on our carrier, shuttled to the very end, and we'll see them again. The protons stay where they are, and they will essentially form a gradient. And again, that will come later, but just keep in mind that this happens all throughout cellular respiration. Essentially, we're going to be stripping glucose down, and we only want the hydrogen atoms, and then we break the hydrogen down into electrons and protons. So at the end of the day, this stuff is all we need for this process. We just need some stripped electrons, and we just need some protons in order to make that ATP. Another thing that happens in this process, we make a little bit of energy by doing this as well. So 2 ATP molecules are generated here. So things like bacteria that can only do this process are able to sustain themselves on this glycolysis action because a little bit of energy is needed, and they're very small, so they don't need a lot, so this is sufficient for their needs. So after all of this happens, we now end up with 2 molecules of pyruvate. So we're no longer glucose at this point, because remember, glucose is a 6-carbon molecule, and we're no longer 6 carbons, but we're 2 3-carbon molecules called pyruvate. So this very first process, glycolysis, breaks glucose into 2 and makes a little bit of energy. We have some electrons and protons and some pyruvate. So these are the 3 outputs that occur in stage 1. Stage 2. We are in the matrix now, folks. So welcome, we're in the matrix. And those electrons and protons are accumulating from stage 1. But the citric acid cycle, also known as the Krebs cycle, and Krebs is just the last name of the scientist who figured a lot of this out, further breaks down pyruvate until it can't be broken down anymore. So this is just further breaking down those 3-carbon molecules, okay? There is an intermediary step right before this happens, but I'm not going to test you on that, where that pyruvate is converted into another molecule called acetyl-CoA. So that describes step 1 here. And again, every time we're essentially breaking down that original glucose molecule, we are just going to be stripping some electrons and some protons. So all we want from glucose are the hydrogen atoms, and then we break the hydrogen atoms down into its counterparts, the electrons and the protons. So every crank, essentially, of this cycle, we're just breaking down that 3-carbon molecule a little bit more and a little bit more until all that's left is carbon dioxide, which we breathe out as waste, and of course what's remaining from the glucose, which is C6H12O6, are those hydrogens, which we then will strip the electrons off of, carry those onwards, and we have some protons building up as well. So we're going to create a gradient with the protons, and we're going to shuttle the electrons eventually. In this cycle as well, we have a little bit of energy being made here. So a little bit of energy, a little bit of ATP, not a lot, but some. And then, of course, we have just some more electrons being picked up from that original molecule. It is at this point that glucose has been completely broken down, okay? We can't break it down anymore because what's left of that molecule, the carbon and the oxygen in glucose, has been breathed out of us as waste, and then the hydrogen that was a part of that molecule has been stripped into its electrons and its protons. So we're done breaking down glucose at this point. And again, all we needed from that sugar were the hydrogen atoms, and we've broken those now. And now in stage 3 is where we actually use all of these electrons and all of those protons that we work so hard into stripping off of glucose. The last stage, and this is, each stage is intricate and complex, but this is where we have a lot of stuff happening. So just take it one number at a time, and you'll figure it out. So the first thing that happens in stage 3, all of those electron carriers that picked up the electrons from the hydrogen atoms that we stripped off of glucose drop them off to a series of proteins embedded in the cristae. And the cristae are the inner membranes of the mitochondria. All right, so that's the first big thing that happens. The electrons are carried off, and then once those carriers have dropped off the electrons, they become oxidized, and that's what it means to essentially get rid of those hydrogen the electrons, so they become NAD+, and that plus just means that they're empty and they can go pick up more electrons. So that's all that means. We can see the black arrow here. NADH is the reduced form, and that simply means it has electrons on it. And then once those electrons are dropped off, it becomes NAD+, and that plus simply means it's able to carry more electrons. So these empty carriers will circle back to the first two processes and pick up more electrons, come back and drop them off, go back and pick up more, drop them off, on and on and on and on. So once our two electron carriers have dropped off their electrons that they picked up from the first two stages, the energy of those electrons, as the electrons are being shuttled from protein to protein to protein, the energy in that process causes these proteins to pump all of those hydrogen ions, or those protons that we stripped from glucose, to form a proton gradient. So you see how these little H pluses, or these protons, are starting to accumulate or form a concentrated gradient on this side of the membrane? That's exactly what we need, because the concentration of protons will fall down a rotor protein and will crank this protein to phosphorylate or hook on a third phosphate group to make ATP. So the energy of those protons falling down this motor, essentially, is what's needed to generate our energy molecule in the form of ATP. So, but why do we need oxygen to live? Okay, so once these electrons have provided the energy to form our proton gradient, which will phosphorylate a precursor molecule into ATP, the electrons have to go somewhere. Electrons cannot be left to their own devices. They would cause too much cellular damage, and they would essentially destroy the cell. Oxygen is waiting at the very end of that electron transport chain to catch them, essentially. So oxygen will catch these electrons and will couple with some protons that are just floating around. And in that process, all three of these products will join together and form water. So oxygen is needed, and it acts as that final electron acceptor at the end of oxidative phosphorylation. Now, I know that was a lot to keep in mind, and a lot is happening here. So just study all four of these steps in the last part. Maybe put them to flashcards, and once you look at the images in the text a couple of times, it will start to make sense. But remember, people, repetition is key. You cannot get this by looking at it one time. There's just way too much going on. So here are all of the steps of cellular respiration in order, and we can see inputs and outputs of each. So this would be a really good image to print off and just look at all of the time. So again, we have our first step, glycolysis, where we essentially break glucose into two, and a little bit of energy is made, and some electrons have been generated or stripped off, and also some hydrogen ions, or those protons, have also been generated. We have our middle step, so the citric acid cycle, and this was that intermediate step that we didn't go through in detail. But this is where glucose is completely broken down at this point, and the carbon and the oxygen in glucose is just released as waste. We've made a little bit of energy here as well, but the main point of this is, of course, to strip the hydrogen of its electrons, and then hydrogen ions are also accumulating all throughout this process, don't forget. And then our last step, all of those electrons that have been stripped off of glucose are shuttled to our electron transport chain. All of those protons that have been stripped off of glucose, will fuel the crank to make ATP, which is what the cell can then use as energy. And in the process, oxygen is waiting at the very end to catch those electrons. It will couple with some free-floating protons and will form a molecule of water that we then breathe out as waste. So water vapor we breathe out comes from this process as well. And we can see that all three of these steps, by far, this last step, or oxidative phosphorylation, makes the most energy. So remember, the goal of cellular respiration is to strip glucose of its electrons and its hydrogen ions to use the energy in those atomic particles to make ATP. That's the main idea here. Glucose is the preferred fuel of the cell, but of course, our bodies are able to switch gears and burn other types of fuel as well. So just keep that in mind. Like if you were to go on a low-carb diet, of course you're not going to be eating sugar, but your body is still going to function, right? So it can switch gears to burn fats and even proteins, different mode of action, but we still get essentially the same result, which is the generation of this ATP molecule that the cell can very easily use to perform its work. But what happens without oxygen? So sometimes that will happen, and we call that an anaerobic condition if no oxygen is present. Like some bacteria, and even in our own cells, this can occur. And that process is called fermentation. So fermentation is anaerobic respiration. So it relies on that first step we looked at, glycolysis. So remember I said glycolysis does not happen in the mitochondria. So organisms that don't have mitochondria, like bacteria, can still live, generate energy because of this process. Now remember, it's very simple in terms of what we learn. We literally just break glucose in half and we have generated a little bit of energy. So for very tiny organisms, that's sufficient. And it will convert that pyruvic acid into something called lactic acid, okay? So this happens for bacteria, and some yeasts as well can do this. And our own cells can do this. So if we're exercising really intensely and we're burning energy faster than we can get oxygen to our cells can switch to fermentation mode very briefly. And that is what causes lactic acid buildup on your cells and what causes muscle soreness the day after you work out really hard. And then, of course, if it's a microbe that is fermenting, we can reap the benefits of that process in our food industry. So this is how a lot of cheeses, yogurts, alcohols, and even other beauty products are made. So the byproducts of fermentation are products that we utilize in our everyday life.